Hey guys, so last little session, I think I paused right here on this slide where it's showing you the structure of the DNA. It was just a refresher for us. So I'm going to quickly go into replication. So far we know that it's a double helix. We know that there's a phosphodiester bond, a phosphate with two sugars, and it forms the backbone of the DNA molecule. The bases are present in the middle. Adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds. Each nucleotide has a phosphate sugar and a base. And we know that we have to start counting the carbons on a sugar from the right side of the oxygen. So the first or 1' carbon has the base attached to it, and the 5' carbon has a phosphate attached to it. Okay, so the next one. Okay, so notice that as I told you before, the phosphate group on the 5' carbon of the deoxyribose sugar. So there's a phosphate group. This is your 5' carbon right here, and the phosphate group is attached on the 5' carbon. But when you look closely, the phosphodiester bond, the phosphate group that is on the 5' carbon of one sugar is linked to the hydroxyl group that is on the 3' carbon of the next nucleotide. So the phosphate group links the 5' carbon of one nucleotide to the 3' carbon of the next nucleotide. Okay, pretty simple. All right, DNA packaging. Why does the DNA need to be packaged, wrapped around so tightly? Why can't it just be floating around in the nucleus? The answer was simple. It's because the nucleus is so tiny. Our cells are so tiny, our nucleus is even tinier, and our DNA, if you stretch it out, it's so huge. Now we have to figure out a way to package it into the nucleus. So if you look closely in the DNA packaging, you have histone molecules, and 8 of them, okay? So 8 histone molecules make up the core of the structure called as a nucleosome. The DNA is wrapped around the nucleosome twice. And this is important, guys. It is wrapped around twice around the nucleosome, and there are 8 histone molecules that make up the core of the nucleosome. The DNA that is hanging out is called as a linker DNA. Okay. All right. What is genomics? Genomics is the science of sequencing, interpreting, and comparing the whole genomes. Where do we use this? We use this a lot in forensic science, and also not just forensic science, but finding out fossils and figuring out our ancestors and a lot more things. So comparing viral DNA to figure out which viral species is infecting us, or which bacterial species is infecting us, and what kind of antibiotics we need to make. So there's a big group of sciences that are really using genomics. Okay, the second point is that only 2% of the DNA is actually coding for proteins, and the other materials do not code for proteins. So out of all the DNA that is present in each cell, only 2% of it codes for proteins. The rest of the 98% is made up of many different kinds of molecules that can be used as regulators of gene expression. They can make up introns, they can make up telomeres, and genes that code for tRNAs or transfer RNAs. Now, in the next slide or so, we'll look at what each one is. What is a regulator? What is the telomere? And what are the introns? So right here I have the types of DNA sequences. The first one is a highly repetitive sequence. So let's say we have a sequence of bases of nucleotides, and you have on the sequence of the DNA you have AA, TT, GG. That's not, that's a normal DNA sequence. But if it is repeating, the bases are repeating themselves quite, for a lengthy sequence is a repetitive sequence. So usually it is composed of 500 to 300 base pairs per repetitive sequence, and there may be 100,000 repeats in a certain genome. Now, if these repeats are clustered in one area, they're referred to as a satellite DNA. And usually these repeats are not clustered, they're dispersed out. So if these repeats, if the bases are repeating and they're clustering it in one place, they're called as satellites or satellite DNA. But usually you don't see them clustering. There was a scientist called as, her name was Barbara McClintock, and she actually discovered that certain genes, especially that are repeating themselves, can move from one location on their genome to a different location, or so from one loci to a different loci. And because they keep moving, they're called as jumping genes or transposable elements. Now, the thing to remember about the jumping genes and the transposable elements, guys, is that they're not actually leaving the genome. Okay, at no point are they detached from the genome. They're in the genome, they're part and parcel of the genome, but they can move from one loci within the genome to another loci, okay? And the evidence of jumping genes was seen in corn kernels. When you see those different colors of kernels on a same, cob of corn, that was an evidence that genes were actually moving and jumping from one loci to another loci in a genome. Okay, she actually won a Nobel Prize for her work in 1983. Okay, let's go to the protein coding genes. These are the ones that we are usually interested in. They're usually single copies, and they produce proteins, and they have to be at the protein producing factories, which are the ribosomes, right? If you remember, a gene is made up of numerous protein coding factors or exons. And these exons are not, a sequence together. Like a genome might have an exon, then an intron, an exon, then an intron, or two exons and one intron, and so on. So they're not always together. They're always dispersed in the genome. So if you remember from last year, introns are not included. Okay, introns are non-protein coding parts of the genome, whereas exons are the protein coding parts of the genome. All right. Structural DNA or structural DNA. I think this should be structural DNA. It is again non-protein coding. It does not code for any protein, and it is usually found near the centromere and the telomere of a chromosome. If you remember the structure of a chromosome, the center of the chromosome is called the centromere, right? And telomere is the end of the chromosome. Now, when it is at the center, it is tightly packed, so it's not exposed to all the enzymes and the proteins that need to be there to help transcription and translation to take place, because it is really tightly packed near the centromere, and that could be one of the reasons why they're not coding for any protein. And telomere, they're at the very ends or the tips of the chromosome, and I am not sure, I don't know why they do not code for a protein. I'll be glad to know if you guys find out. Okay. And then we have pseudogenes. pseudo standing for false. So they seem to have, be looking, they seem to be genes coding for proteins, but they actually do not code for any proteins at all. Scientists believe that maybe at one time they had the protein coding function, but they have lost it. Now they don't code for any proteins anymore. That's again a theory which has not been proved. Okay, so now this little chart right here gives us the different kinds of genomes that we just went through. The protein coding genes, also called as exons, make up about 2% of the entire genome. The introns are the non-protein coding genes. These are actually interspersed within the genome along with the exons. It makes up 24% of the genome. So exons make up to 2%, introns 24%. The highly repetitive sequence is the highest frequency. It has 45%. It makes up to 45%. The structural DNA makes up 20%, pseudogenes about 2%, and others 7 to 8%. Okay. Short tandem repeats, or they're generally called as STRs. This is very, very interesting, especially in profiling subject or a person. It is believed that human beings have certain regions in their genome that are very short and they're, repeats. That means certain bases are repeated in a particular sequence. And this is the sequence that differs from one person to another person and makes you unique and different from the next person. Okay. That's why they're called, they're very important in DNA profiling. So they're short, specific sequences that are found in the genome. And this is where one person's genome differs from the next person. So short tandem repeats are the ones that usually scientists look at in order to know and figure out if this is the right person that they're wanting to pursue or apprehend, or if this if this particular person has this particular STR, then how does it link to a certain disease? Is this common in people with certain kinds of diseases? So there's a lot of help from short tandem repeats. All right, let's come to DNA replication. You all, I hope you remember that DNA replicates by a semi-conservative method, and the two scientists that worked on this method and confirmed that DNA replication is semi-conservative were Matthew Meselson and Franklin Stahl. And if you remember, we did in class a hands-on activity with like pipe cleaners to different colors of pipe cleaners to see how DNA can replicate, conserve half of its originality and then make a new one. Well, Meselson and Stahl, they used heavy nitrogen. Okay, so they grew bacteria in a culture that had heavy nitrogen, N15. So a normal nitrogen would have N14, would be N14, but heavy nitrogen is N15. And then they grew this bacteria that was grown in a culture of N15. They took it and they grew a second generation in a culture that had the normal light nitrogen. Okay, and this is what they noticed. They noticed that half of the DNA strand had N14 in it, and the other had N15. And in the next generation, they found that there was one completely new DNA that was just N14, and there was another one that has half N14 and half N15. Okay, and so this led them or help them prove that yes, DNA replication is semi-conservative. One half is conserved and the other half is made new. Okay, so we knew that already. Let's see what we didn't know. Okay, bacterial DNA, it is circular, and it does not need to be wrapped around histones because there is, it does not need to be packaged into a nucleus because bacteria is prokaryotic and there is no nucleus in the bacteria. So the DNA is just hanging out, so no packaging, no histones necessary. It's big and it's circular, but it's not as big as the DNA of a eukaryotic organism. Each cell of the eukaryotic organism has its own nucleus and a large amount of DNA. Okay, that each cell has more DNA than the bacterial DNA. So, and it is not circular, it is linear. Because replication has to happen fast and you don't have forever for your DNA to replicate. In eukaryotes, replication starts off at many different locations, okay? These locations where replication starts off are called as origins of replication or origins of replication. So it begins at a bubble. And if you remember, the enzyme helicase is the one that unwinds the DNA, and it unzips it, also breaking the hydrogen bonds between the bases. So now the bubble that is formed, because it starts off at many locations, many bubbles can be seen on the DNA strand, and it extends in both directions. The bubble extends in both directions. Okay. The extension is also called as the replication fork. So you see the fork opening up in both directions, originating from the bubble. All right. Okay, so this is a little bit more information, but I'm sure you can handle it. Let's look at it. Let's look at what we know first. We know that helicase is the enzyme that unwinds the DNA and unzips it. We know that DNA polymerase is the one that adds new nucleotides, and we know that ligase is the one that adds up the newly formed strands. The strands on the lagging strand are called as Okazaki fragments, and then you have one leading strand. We know this so far. Okay, let's just look at a little bit deeper and learn a bit more. Once the helicase unwinds this double-stranded DNA, there are proteins called as single-strand binding proteins that go and bind to the single strand. Okay, what is their purpose? Their purpose is to prevent these two strands from rewinding again. Okay, so the single-strand binding proteins keep those unwound strands separate and prevent them from rewinding again. Then, if you notice, there's an enzyme called as primase. What do you think its function is going to be? It's going to add a primer. Okay, primase adds a primer. Now, primers are made up of small RNA nucleotides. Okay, the primer is made up of small RNA nucleotides. How do you relate this? Whenever you're painting a room, all right, let's say you're painting a room, and if you remember, sometimes, I hope most of the times, you actually put a coat of the primer first, and then you put a coat of the paint. So the primer is actually helping the paint to stick on the walls better, right? So try to connect that to this primer right here. So primase is the enzyme that adds the primer, and the primer acts as a precursor for the DNA nucleotides that need to be added in the growing new strand. All right. And remember, DNA nucleotides are always added from the 5' towards the 3' direction. That's the direction that the DNA grows in. So the primase adds the primer, then the DNA polymerase 3 starts adding nucleotides on it, okay, onto it. DNA polymerase 1 comes in and it removes the primer. Once the primer's function is done, remember a primer is made up of RNA and not DNA. So once the primer's function is done, the DNA polymerase 1, so you guys have to remember there's two kinds of DNA polymerases that I'm talking about. Talking about DNA polymerase 3 that adds the DNA nucleotides, and DNA polymerase 1 that removes the RNA primers. So if I talk about 3 and 1, there must be a 2 somewhere, right? And we'll figure that out pretty soon. But so far in replication, we are using DNA polymerase 3 that is adding new DNA nucleotides and DNA polymerase 1 that is removing the primer once its function is completed. Then comes the enzyme DNA ligase, and we know the function of ligase is to attach the growing Okazaki fragments together, okay, to link them together. Anything else? Am I missing anything else? Just make sure you remember that in the leading strand, we don't need primer to be added so many times because it is the leading strand. So primer is added once at the 5' end towards the 3' end. So 5' towards the 3' end, primer is added once. So DNA polymerase has to act once and remove that primer. And then the leading strand, the DNA polymerase 3, just keeps on adding new nucleotides to it. Good? All right, here. So let's look at the summary of the enzymes and what their functions are. So helicase unwinds the double helix. Primase synthesizes the RNA primer. Polymerase 3, new strand, it adds the nucleotides, DNA nucleotides in the 5' to 3' direction. Polymerase 1 removes the primer, replaces it with DNA. Oops, I forgot to mention that. So I kept stressing that DNA polymerase 1 removes the primer, but that space has to be filled in with the DNA, right? RNA is removed and you have to fill in that space with DNA. And DNA polymerase 1 takes over that function as well. Okay, and then you got DNA ligase, joins the ends of the DNA and the for the Okazaki fragments of the Okazaki fragments on the lagging strand. All right. Now, all right, guys. You remember bacteria, it replicates every 20 minutes, right? So replication has to happen at a very fast rate. And it is said that about 4,000 nucleotides per second, okay, are replicated. 4,000 nucleotides per second. But if it's going that fast, don't you think there'll be some mistakes made, wrong nucleotides being added? Maybe ligase is not able to attach all the Okazaki fragments. Maybe the RNA primer is not being removed by the DNA polymerase 1. Many things could happen, and it's millions and millions of nucleotides that keep going on, and a big, nucleic acid that we have. But it's remarkable to notice that DNA has a self-check mechanism. So every time it replicates, it proofreads itself and self-checks. And there are different enzymes that do that. For now, just remember that DNA has a mechanism where it proofreads itself. So before it, confirms that, yes, this is a good copy of myself, it proofreads it. Okay. And also wanted to tell you that that's the reason we are not all mutants. Okay. So that's the reason we are not all walking around with something or the other going wrong in our DNA, and that's being expressed, or an essential protein that's not being expressed, and that's why we look weird, because of the remarkable capacity of the DNA to check itself, to proofread itself. Okay. All right. We got to know about this method that a scientist called Sanger came up with to sequence the DNA. Okay, so this is a method that was done by Frederick Sanger in 1970, and there's a link to the video. Sorry, there's a link to the video. I recommend that you guys watch the video before you read further, because that'll help you kind of visualize what I'm talking about. Okay, so just take a minute and watch that video, guys. All right, you might in that video, what they do is they have, let's see, 4 tubes. In that video, you notice that there's 4 tubes. Okay, and each tube, excuse me, has a different dideoxynucleotide triphosphate. Okay, now dideoxynucleotide triphosphate, if you look at this picture right here, we know that the sugar, the ribose sugar in the DNA is a deoxyribose sugar. That means the second carbon, 2' carbon right here, did not have an oxygen, so it only had a hydrogen. That's why it was a deoxyribose sugar. But in a dideoxyribose sugar, you're wanting to remove 2 oxygen molecules. So di meaning 2, deoxy is lack of oxygen. So even on the third 3' carbon right here, you see an OH. But if you look at this 3' carbon, the O is removed and you're left with only H. Okay? And I'm just saying it is dideoxynucleotide triphosphate because this base right here, what they did was in one tube they had adenine, so it would be dideoxyadenine triphosphate, and in another tube it would be dideoxyguanine diphosphate or triphosphate, and the third it would be dideoxytyrosine triphosphate, and then cytosine triphosphate. Got it? Now, why is it so important? So what if we took away the oxygen on the 3' carbon? So what, right? So if you notice, phosphodiester bond, guys, if you remember, the phosphate group is formed between the 5' carbon of one nucleotide and the 3' carbon of the next nucleotide. But go back, what did we do? We took away the oxygen on the 3' carbon. If you take away the oxygen on the 3' carbon, my phosphate group, which is usually what it does is links the phosphate group of one sugar on the 5' carbon sugar to the hydroxyl group on the 3' carbon sugar. It does not have a hydroxyl group anymore. It only has hydrogen. So that bond does not form. So DNA does not grow. It just, whenever it encounters that particular dideoxynucleotide, it just stops. All right? So that is the purpose of that dideoxynucleotide. So knowing the structure and knowing, having that knowledge helped Sanger to create this amazing method where he could actually sequence out the DNA and figure out, what the bases are in a nucleotide, in a nucleic acid strand. So that's where I'm going to end. I really hope you watch this video first before you figure out the Sanger's method, guys. All right? So this is where the session ends. I hope I will see you with questions in the classroom. Have a good day or night.